to host a local colleague, actually a new addition to our liver center, Dr. Anna Arruda. She's a new assistant professor in the Department of Nutritional Sciences and Toxicology at UC Berkeley. She's originally from Brazil. She got a bachelor in biological sciences at the State University of Sao Paulo, and then followed that up with a master's and PhD degree in biochemistry at the Federal University of Rio de Janeiro. She then came to the US and did a postdoctoral fellowship in Boston at Harvard School of Public Health. In the process, she obtained a prestigious fellowship by the Pew Charitable Foundations. And she then started her own lab actually earlier this year at UC Berkeley, and is being supported by a Chan Zuckerberg BioHub investigator grant. She has more than 20 publications so far, so quite a bit, including in prestigious journals like Nature and Nature Metabolism. And her work revolves around studies of how organelles, in particular ER and mitochondria, adapt to nutritional stress to maintain metabolic homeostasis, and on the basis of that, how organelle dysfunction leads to fatty liver disease and diabetes. So we're very excited that she made the trip over the bridge, and really looking forward to her talk. And actually I have to correct myself, we now do have the presentation on the screen. Okay. Welcome.
Okay, so do you hear me? Okay, thank you, sorry. Okay, so the main focus of my lab, as I was saying, is trying to understand mechanisms involved in the development of metabolic diseases such as type 2 diabetes, fatty liver disease, and obesity. And we take the perspective of organelle biology. So I wanted to start by talking a little bit about metabolic adaptation. So in general, nutrient intake during cycles of feeding that we all undergo every day. So nutrient, when it comes to our organisms, usually causes a mild stress to our cells, organelles, and metabolic networks. But this is rapidly overcome thanks to the capacity of our cells and tissues to activate adaptive systems and reestablish homeostasis. So the problem is under chronic overnutrition, what happens in obesity, the chronic consumption of excess nutrients and calories over time promotes a continuous stress to the cells, tissues, and organelles, which is higher both in amplitude and also in frequency. And over time, the adaptive systems start to get exhausted and start failing until a situation of unresolved distress takes place. And this is usually characterized by accumulation of toxic products of metabolism, such as RAS, unfolded proteins, toxic lipids, which then triggers more general forms of stress response, such as metabolic inflammation, and then leads to metabolic deregulation, such as insulin resistance and dyslipidemia. And this turns into a vicious cycle that, if it's not controlled at the correct time, will certainly lead to development of metabolic diseases, such as this mentioned here. So an important aspect of metabolic adaptation occurs at the level of the organelles, for example, at the mitochondria or the endoplasmic reticulum. And this is because the organelles, they are key parts in receiving the nutrient signals, and they compartmentalize the metabolic reactions, and they host actually many of the main metabolic enzymes. And in situations of metabolic diseases, they get dysfunctional. So the question you're trying to address is how a chronic intake of nutrients in excess leads to organelle dysfunction and failure of the organelles to communicate with each other, and whether we can target metabolic adaptation at the organelle level to improve metabolic health. So now I'll focus a little bit more on the ER. So the ER is a very diverse organelle, both in terms of function and also structure. So although it's formed by a single membrane that is connected and is spread throughout the cytosol, it presents itself with distinct subdomains. For example, the ER sheets that I can showing here in this EM, which is decorated usually by the ribosomes, also called the rough ER. And this subdomain of the ER is dedicated to functions related to proteostasis, such as protein synthesis, protein folding, and secretion and quality control. The ER is also formed by a network of tubules that is usually devoid of ribosomes, also known as the smooth ER. And the tubular ER seems to be more related to lipid metabolism, lipid synthesis. This is also the sites of lipid droplet formation. And this domain of the ER is also able to form contact sites with every other organelle in the cells. And another aspect of ER function that is important for my work later on is that the ER is the main site of calcium storage in the cell and participating calcium signaling in the cells. And it's interesting to see that the proportion of these morphological domains of the ER sheets and tubules, they vary a lot according to the functions or the metabolic functions of a certain cell type. For example, in the pancreatic acinar cells, which are cells dedicated to the secretion of digestive enzymes, so it's a very secretory cell, the ER is mostly found as ER sheets, rough ER sheets, as shown here in this electron microscopy, and this fills out the entire cytosol. And the other side of the spectrum are the Leydig cells, which are cells specialized in steroid hormone synthesis or lipid hormone synthesis. And in these cells, the ER is mostly found as ER tubules, as you can see in this EM. And then in the middle are cells such as the hepatocyte or also the enterocytes, which are more pleiotropic cells. So in addition to be highly secretory, so the hepatocyte secretes a lot of protein, lipoproteins, but is also heavily involved in lipid synthesis. And in those cells, you can find the ER in the forms of ER sheets and also tubules, which is hard to see here, but I will show later on with higher resolution. And in those cells, the ER is more dynamic. So this proportion or this morphology changes according with the metabolic states of the cells. So this is just to illustrate that the structure of the ER is very much related to the metabolic demands of the cells and important for the function of the ER. And given the importance of the ER in the cells, this organelle exhibit a high plasticity, which means that the ER is able to adapt to a variety of forms of stress in the cells. And it does so because it hosts very strong adaptive systems, for example, the unfolded protein response, which I'm not going to go in detail here, but this is a response that senses the ER stress in the ER lumen, or like the folding of the proteins in the ER lumen, and when ER stress is detected, so this adaptive system is triggered to recover ER homeostasis. However, even with these strong systems, under chronic overnutrition, the ER cannot maintain homeostasis. So data for more than 20 years now have shown that the ER presents chronic dysfunction in several metabolic tissues under obesity, for example, the pancreas, the hypothalamus, adipose tissue, and the liver. And actually, in this condition, the unfolded protein response is defective or is presented in very unusual and ineffective ways. And then the question is why is ER cannot handle stress imposed by metabolic conditions. And at least in the liver, it seems that one important and early problem that happens in the ER is that this organelle cannot maintain calcium homeostasis very well. So in this paper published by Rotem lab, where I did my postdoc, what they did, they isolated ER from lean and obese animals from the liver. And then they performed lipidomic analysis, and they detected that obesity leads to changes in the phospholipid composition of the ER membrane. So the ratios of PC and PE are altered, and these affects of the enzymes that sits on the ER membrane, such as SERCA, which is a calcium pump that pumps calcium from the cytosol to the inside of the ER. And calcium is an extremely important ion for ER function because it controls chaperone activity. So if you have lower calcium, this will affect protein folding. So this seems an early event happening in obesity or in fatty liver that may trigger ER stress. And later, we also showed that alterations in calcium signaling not only affects the ER itself, but also affects other organelles such as the mitochondria. So very briefly, in this study, we performed electron microscopy in the liver sections from lean and obese animals. And here the model we use is the OB-OB, or the leptin deficient mouse that becomes morbidly obese. And what we notice is that while there was a change in the morphological organization of the ER in the hepatocytes, and this was characterized by more frequent connections between ER and the mitochondria, as I'm highlighting it here. And just to make a long story short, what we discovered in this work is that these excessive connections between the ER and the mitochondria, which happens in the obese liver, leads to excessive calcium flux from ER to the mitochondria through channels called the IP3 receptors. And then mitochondria becomes calcium overloaded, which triggers ROS production and activation of stress signaling protein molecules such as JNK, which then leads to problems such as insulin resistance. So this work showed that ER stress is tightly linked to the mitochondrial stress through this calcium flux between the organelles. And if we uncouple this phenomena, for example, by knocking down the IP3 receptor at these junctions in the hepatocytes, we see that this leads to improved mitochondrial function, also improved ER function. And this has systemic consequences, for example, improvements in glucose tolerance in animals where we downregulated IP3 receptors. And we later on another work, it was shown that deletion of IP3 receptor in the liver in the context of obesity, fatty liver, also improves the steatosis in the livers, as you can see here. So this shows that targeting mechanisms that control intracellular calcium flux in the organelles, the ER and the mitochondria, for example, SERCA or the IP3 receptor, they result in better metabolic homeostasis and better organelle function. So maybe thinking about molecules that can regulate these enzymes and channels could have a therapeutic relevance for fatty liver disease or obesity-driven fatty liver disease. Okay, so after this initial work, we became really interested in this fact that obesity leads to this alteration in the ER morphological organization in the liver, especially because of the type of the changes in the ER structure we are observing in the obese animals was very different than what we would expect in situations of ER stress. So for example, when you induce ER stress in cells with chemicals such as tunicamycin or tapsigargin, this leads to expansion, dilation of the ER. I don't know if it's clear here, but also it leads to ER proliferation. And this is a homeostatic response to increased protein folding. So this was very different what we are seeing in the obese, although we do see ER stress in the obese hepatocytes. So we wanted to understand this in a deeper level, how obesity impacts hepatic ER structure organization in the liver. However, at the time, the techniques we had, so the transmission electron microscopy and confocal microscopy was not enough to fully resolve the ER structure. So to do this in the native tissue environment, we use a different technique, which is called focused ion beam scanning electron microscopy. And we did this in collaboration with Harold Hess and Shan Shu's group from Genelia. So briefly, this technique works by taking a scanning electron microscope image of the surface layer of a sample. And then with a focused ion beam, the surface layer is removed from the sample and then reveals the layer underneath. So another picture is taken, and it goes like this until the entire volume of the sample, in the case of liver volume, is imaged, and the entire sample is also consumed during this process. And this type of microscope gives the highest spatial resolution that is achievable so far, so around 2 to 80 nanometers isotropic resolution. And for each data set, we generate a lot of pictures. For example, in one data set, you generate 6,000 to 8,000 pictures, terabytes of data. And then the next challenge is to annotate this data to see what is an ER in this figure, what is a mitochondria or other organelles as well. So this would be impossibly doing manually. So for doing that, then we generated a pipeline based on machine learning automated segmentation, using a convolutional neural networks, and we are able to achieve very high precision in organelle segmentation using this pipeline. So here I just wanted to give you an example. So this is the reconstruction of the EMs from the liver from a lean mouse in the fed state. So here we are going in Z, and you can see the ER structure that's annotated here in blue, and then the mitochondria annotated here in purple, also showing the mitochondria cristae. So this is a very small portion of the data set. So the data set covers many cells at the same time. And then I wanted to show in this video the scope of the annotation that we did. So here again, the ER is in green, the mitochondria, we can see the nucleus here. So multiple hepatocytes were imaged in each of our samples. So this again is from the lean mouse. So we did this type of analysis using livers from normal lean and then obese, OB, to started to do a quantitative comparison with these different structures or subcellular structures of the liver. So here's just to show the reconstruction of the liver from the lean and then the obese. So as expected, the obese has a much more lipids droplets in the hepatocytes, which characterizes the steatosis. And then to quantify the organelles, we extracted all the organelles in 5 different cells, full hepatocytes that are inside this liver volume. So here is just one example of all the mitochondria that are contained in one hepatocyte. And this is all the ER network in one hepatocyte. And then we focus on the ER to compare the ER structure organization, comparing lean and obese liver samples. So here is the section of the one hepatocyte from the lean wild type. And as you can see here in blue, in the lean condition, the ER is predominantly found as organized as these stacks of parallel organized ER sheets, as you can see here. There are also some tubular network in this hepatocyte. But when we did similar analysis with the obese hepatocyte from the obese animals, you can see that there was a massive decrease of the ER organized in stacks of ER sheets, as you can see here. And instead, mostly of the ER was found as ER tubules, as we can see here. And we couldn't see this when we did 2D TEMs. We were actually thinking there was less ER in the obese, but in fact, there are actually similar ER amounts, but it's just there's a remodeling in the structure of the ER. And then in order to quantify these structures, we resegmented all our data set, now separating the ER tubules from the ER sheets. So here the ER sheets are highlighted in red and tubules in gray. And then we quantified them and found that in fact the ER sheet to tubule ratio is dramatically or like significantly decreased in obese condition. And then the question is, but what is the functional implication of such remodeling of the ER structure for ER function or for the hepatocyte metabolism? So as I mentioned to you in the beginning of the talk, the ER sheets are the preferential site for protein synthesis, protein translocation, and folding, and also protein secretion. And this is because the flat surface layer of the ER sheets is accommodate large structures such as the polyribosomes, which binds to the SSRP receptor in the ER membrane. And also the ER sheets are the main location where the translocon complex and accessory proteins are expressed. So the nascent polypeptide that is exiting the ribosomes, entering through the SEC61 or the translocon complex, and is transported across the ER membrane and is folded in the ER lumen. And this preferentially happens in the ER sheets. So we also checked the expression levels of the translocon components, which will be important for translocation of the peptides from the ribosome, exiting the ribosomes to inside of the ER. And we saw that SEC61, beta alpha, and TREP alpha, which is an accessory protein of this translocon complex, are also decreased in obesity in accordance with the decrease of the ER sheets. So we would suppose that the process of protein folding or protein translocation and secretion would be affected in obese condition. So to measure that, we, or some aspect of that, we use a reporter assay that also we developed in Gokhan's lab, Gokhan Hatamichli's lab. And this assay is based on the folding and secretion of a protein called ASGR. So this is a slow folding protein. And in this construct, we fused the ASGR with a luciferase, which is called C-loop here. And in the same construct, we express another luciferase called G-loop under the CMV promoter, which will serve us for control for expression levels of this construct. So the premise of this assay is if the cells of the ER is in a low stress or like a normal healthy condition, so the ASGR will be synthesized, translocated inside the ER, folded, and then secreted. And we can measure the luciferase signal in the media. However, if there is high stress, then we expect to see lower amounts of signal of secretion of this protein because there will be problems with the folding and secretion. So we use this assay, we clone this in adenovirus to be able to deliver to primary hepatocytes, and then we isolated primary hepatocytes from lean and obese. And then what we saw is that, in fact, in obese hepatocytes, there was a lower levels of C-loop to G-loop ratio in the media, so which means there was less protein folding and secretion based on this assay. So then the next question we asked is, can we restore ER structure organization in order to improve ER function? So the question here is, can we target the form of the ER to change the function of the ER? And would this have a metabolic consequence for the cells? So to do that, we then turned our attention to the ER shaping proteins. So the shape of the ER is maintained by the relative abundance of the ER shaping proteins, and also how these ER shaping proteins are connected with the cytoskeleton. So the ER sheets are shaped by a protein called CLIM63, which is a transmembrane protein that oligomerizes in the ER lumen and forms a spacer between the two ER membranes, maintaining the ER membranes in a constant distance, as shown here. And another protein that has been shown to regulate the flatness of ER sheets is called the P180. The ER tubules are maintained by proteins that contains a special domain called the reticulum homology domain, which such as RIP5 and reticulons. So the reticulum homology domain corresponds to these short hairpins that are inserted in the membrane, but it doesn't cross the membrane bilayer, so bending the membrane, forming the tubulation. So it's been shown before in cell lines by a group from Tom Rappaport, Gia Volz, and others, that if you overexpress CLIM63, you drive ER sheets at expenses of ER tubules. On the other hand, if you overexpress RIP5 or reticulons, you make the ER in forms of tubules. So use these tools in order to try to reshape the ER in vivo condition. So here, first I will show the data we obtained when we try to restore the ER sheets, and here we use CLIM63 for that. So first we wanted to see if in vivo in the liver we could recapitulate the data that we see in cells. So we also use adenovirus to overexpress the CLIM63. and added this in OB animals. And we did the same pipeline with the FIB-SAM and reconstruction of these the livers obtained of the obese animals overexpressing CLIMP63. And we saw that, yes, when we overexpress CLIMP63 in the obese mice, we were able to induce proliferation of ER sheets in these hepatocytes. And here, this is a transmission electron microscopy. We also saw that the ER sheets that are resulting from CLIMP63 overexpression, they show the presence of ribosomes attached to the ER sheets, as you can see here. And this is just the quantification of this data. So we were able to restore partially the ER sheets in the obese animals by using CLIMP63. And then we asked, but what would be the consequence for ER function or for hepatocyte metabolic status? So here we use the same assay that I described previously, the ASGR, to see whether there was a change in the ER folding capacity when we increase ER sheets. So here again, I'm just repeating the data showing that in obese, there is a decrease in the folding capacity of the ER based on ASGR folding. And when we overexpress CLIMP63 in the obese hepatocytes, we had a partial, we have an increase on the, this ER folding capacity. We also analyzed many aspects of ER function. One of them also, we checked for the proteins that are downstream of ER stress, for example, ATF, or like integrated stress response, such as ATF4 and CHOP, that are constantly upregulated in obese condition. And we saw when we overexpressed CLIMP63, ATF4 was decreased, as well CHOP, as you can see here, showing that probably the ER is in a better condition when in the form of ER sheets in this situation. And then we also check for some of the metabolic function of the ER and also the cells themselves. So here we did an assay to measure lipogenesis based on 14C acetate incorporation into lipids. And we see that overexpressing CLIMP63, again, here was in hepatocytes, primary hepatocytes again, we saw a decrease in lipogenesis, and at the same time, we saw an increase in fatty acid oxidation. So the lipids were being less produced and more burned. And as a consequence, we saw that the liver steatosis, the accumulation of the lipid droplets in the liver of the obese animals after overexpression of CLIMP63 was decreased, as you can see here. Although these animals, there was no difference in body weight. They show the same body weight, but a better management of the lipids in these livers. And then in the final part of this work, we asked the opposite question. What if we just induce ER tubules in a normal mouse? Would this resemble some aspects of ER dysfunction or metabolic alterations that are present in obese? So this was not so easy to do. Like, we used several of the RIP isoforms and the reticulone isoforms to turn the ER from sheets and enrich in ER tubules. So here I'll show one of the tools that gave us positive answers for this question. So here we overexpressed RIP5, and as you can see, there was a huge proliferation of ER tubules in the hepatocytes. We never saw this before, of course, not in a normal, but it's very clear. And then what we saw, also, like, we saw that in these animals, we see lipid droplets around these ER tubules, as you can see here. So we did some H&E staining of the livers from the lean mouse, and we saw higher accumulation of lipid droplets when we proliferate ER tubules with RIP5. Also, the triglyceride content was higher. And then a paper published a little bit earlier have shown that when they induce ER tubules in cells, the tubules facilitates the nucleation of the TG in the ER membrane. It facilitates the budding of this TG in the form of lipid droplet. So it decreases the energy that is necessary for the TG to bud and form the lipid droplets. So we think that could be an explanation why when we turn the ER into ER tubules, there was a higher amount of lipid droplets formed in those in the tissues. So the summary of this work is that then the hepatic ER structure is responsive to nutritional stress. So in obesity, there is this transition from ER sheets to ER tubules. And we also saw more of the ER mitochondria contact sites, which we are now quantifying in our 3D data sets. And that also it's possible to target or to change ER shape to alter ER metabolic function. And this has an impact in the cells. And in the next few minutes, I also wanted to show some of the work we are doing in our lab right now. So this work opened a lot of questions, like in terms of the obese, we are trying to understand how exactly the change of the ER shape results in alterations in metabolic function, for example, lipogenesis or fatty acid oxidation. But one aspect that is in our mind right now, so everything I showed to you right now is exploring how a pathological conditions such as obesity leads to ER changes in ER shape or ER remodeling. But I wanted to ask the question, is this ER remodeling also part of a metabolic adaptation in physiology, in the normal cell condition, when the hepatocyte needs to change function to respond to different nutritional demands, for example. So the paradigm we are exploring now is fasting and feeding, whether the metabolic adaptations required to maintain these conditions also are involved changes in the ER structure. So we are using lean animals, and then one group is in the fed condition, and another group we do overnight fasting, around 16 to 20 hours fasting, and we are analyzing what's the impact of this to the ER subcellular organization, and also the mitochondria as well. So as you can see here, this is just the 2D TM from the fed hepatocytes. So the ER, you see the ER sheets here. So there are some ER tubules that's not so easy to see in the 2D EM. And then when we fasted these animals, one very noticeable thing is that the mitochondria becomes much more bigger and elongated. And this has been shown before to increase fatty acid oxidation capacity during fasting. So here you can see the 3D reconstruction of the mitochondria. It really becomes flat, elongated, and fused. And we also saw changes in the ER morphology. So what we observed here is that the ER sheets changed the configuration from these stacks of ER sheets to the ER sheets are curved and around single and around the mitochondria, as you can see here. And we actually noticed that the distance between the ER and the mitochondria in the fasting condition is higher than what was shown for the MEMS, the ER mitochondrial contact sites, which are very tight and formed by the smooth ER. So in fasting, what we see is the rough ER sheets that go around the mitochondria like here. And then we did actually a time course and quantified the amount of ER covering the mitochondria perimeter, and we see that the more fasted the animals are, more you have this, what we are calling now envelope ER, where the ER is enveloping the mitochondria. So here is just like a small volume of the liver of the fasted condition. So here you can see the mitochondria. So the ER around the mitochondria here we highlighted in white. So you can see the ER really forms a scaffold for the mitochondria in the fasted condition. And I just reconstructed one mitochondria so that you can observe. So this is one mitochondria from the fasted liver. This is why it's flat and fused like this. And this is all the ER around the mitochondria, what we are calling envelope ER. And we don't know the function and what are the molecular structures that defines this type of ER contact sites. And just to finish, one more thing is that the liver, as you know, is not a homogeneous tissue. It differs according with the zonation, according to the proximity of the hepatocytes of the portal or in the central vein. The hepatocytes have very different metabolic functions in the distinct areas of the liver. So we try to see whether these changes in the ER sheets are homogeneous across the liver or relates, or it's different across the pericentral, periportal areas. And what we see is that this formation of this envelope ER in the fasting is more pronounced in the periportal and mid-lobular areas, but it doesn't happen so much in the pericentral. So maybe it is related to functions around this areas. For example, one function that seems to be higher in periportal hepatocytes is fatty acid oxidation because of the higher concentration of oxygen. So we are thinking whether fatty acid oxidation requires also the remodeling of ER around the mitochondria. So these are some things that we are exploring right now. And I think I'm still, yeah. So the questions we are doing is what are the molecular mechanisms that are involved in ER remodeling, in fasting? So we are looking to whether the shaping proteins are modulated or whether there is a change with the interaction between the shaping proteins with the cytoskeleton during fast feeding and obesity, and how precisely ER shape changes ER function. So with that, I wanted to thank Gökhan Haftamishli's lab, where I did my postdoc and stayed many, many years in Gökhan's lab, and he was outstanding mentor for me and a friend and a colleague, and all the people that I met there. Our collaboration, Ingenielia, Harold, Shang, which are collaborating with us now in my independent lab, and our lab. So Gunesh Parlaku, which he did all this work we did together. He's now also working in our lab here in Berkeley. And I'm starting the lab, so I was able to hire some people already and start doing experiments. And yeah, I hope to follow up next year to show what we will produce there. So thank you very much for your attention, and I'm happy to answer any questions.